All right, guys, so we're going to be looking at, in terms of forensics, how we analyze DNA and information that we can get from DNA. Blood and bodily fluids are the most common evidence that get used for testing DNA. Like I said, this might be blood, but it could also be saliva, it could be urine, it could be someone blew their nose in a tissue, so it could be from snot, it could be from semen, other things like that. Where we're looking at the blood, remember blood has 4 different parts. So it has our red blood cells, which are our oxygen carriers, our plasma, which is the fluid that carries cells around our bodies. We have the platelets that aid in clotting, and then we had our white blood cells, which are our immunity. So they defend against infections, foreign antigens, things like that. One thing is our red blood cells do not actually have a nucleus. So red blood cells do not contain DNA. They're one of the only cells in our body that do not have a nucleus. Think of, in terms of biology, that also means they cannot replicate themselves. The red blood cells have to be produced elsewhere within our body. However, our white blood cells do have a nucleus and contain DNA. So whenever we are testing for DNA, we're actually testing those white cells is what we're going to be looking for. And whenever we have even a single drop of blood, that can have anywhere from 7,000 to 25,000 white blood cells. So even though we do not have a large amount of blood and we have a very small sample, there's still a lot of cells for us to test for DNA. So only a few white blood cells are needed for us to extract DNA from a sample. And part of that is because a lot of DNA will undergo PCR, which is polymerase chain reaction. This is a lab technique used to make multiple copies of DNA for testing. So this is a machine that they put it into, and the machine, they actually add in different bases and other things, and it will take the one strand of DNA and then it will make 2 copies of that DNA. Then those 2 copies can be made into 4 copies. Those 4 copies can be made into 8, and it will continue this until it has thousands and millions of copies of that sample of DNA. And I should mention it makes copies of the DNA because it amplifies it. Since there's so much of the DNA, once it makes copies, it's easier to test for things. So with PCR, that tends to be a step in the DNA fingerprinting or profiling. And this can be useful for different purposes. Ways that these tend to be used are to identify potential suspects whose DNA could match evidence left at crime scenes. Because the DNA strand is millions of base pairs long, what we tend to do is we tend to look at different segments that tend to be different from one human to the next. And we look to see who has matching segments. And that can kind of help narrow down our suspect field a lot of times. This can also be used to clear a person wrongly accused of a crime. So if they do not have the same DNA segments as the DNA at the crime scene or on our victim, then they could not have left that blood at the scene. These can be used to identify crime and catastrophe victims. Sometimes victims are not physically identifiable. They might have been burnt, the body damaged, things like that. Or if we simply have a lot of bodies in like mass graves and stuff, they can use those to identify individuals. Because a lot of times if we don't have their DNA to test on file, we might have relatives' DNA that would have similar markers. These can also be used to establish paternity and other family relationships. So they would look at a child would have half of their markers from their mother and then half of their markers from their father. So they could eliminate people that could not be their father based off of if they do not have similar markers. This can also be used to match organ donors with recipients in transplant programs. So we talked about with matching up blood type, but with organs, there's other things that come into play, other antigens, other proteins that they want to make sure the body is not going to reject. So they will try to find as many similarities as possible when matching up those organs. And a lot of times samples collected at a crime scene are going to be examined to determine if they are appropriate for DNA analysis. We may be looking at, is it fresh blood? Has it been damaged or degraded in any way? And it's possible that it wouldn't be able to be analyzed for DNA if it's too old, if all the cells have ruptured and things like that. For a sample to be tested, it has to be properly prepared. So DNA has to be removed from whatever object it's on. Now, if we have blood on the floor, we can simply swab it and collect it. If it's blood on like a shirt, we would have to soak it to get the blood to detach from the shirt, essentially. The DNA then has to be extracted from the cell because we don't want the cell and all the organelles. We just want the DNA itself. So to isolate the DNA, the cellular components have to be removed. So they will remove all the fats, proteins, and carbs that are within the sample, as well as any other organelles and things like that. Once all cellular components are removed, enzymes are used to release the DNA from the chromosomal packaging. So if you remember, chromosomes are very tightly wound, where the DNA is, wound around to make those X's. They need that to unwind. So then it will end up stretching out into these long strands. And remember, enzymes, we talked about these in biology and in chemistry. Enzymes are a type of catalyst. So enzymes are proteins that cause a chemical reaction to occur at a rate that's sufficient to support life. So it basically makes the chromosome separate and spread out faster than if we had just left it alone to do by itself. And once the DNA is extracted, now we can characterize it and test it. So part of the process of characterizing and analyzing that DNA is it will go through RFP analysis for DNA fingerprinting. So this is going to be a process for DNA fingerprinting. RFLP is where we have restriction enzymes are used to cut the DNA into fragments, into smaller pieces that are different lengths and different polymorphisms, so different shapes. So that's where the letters RFLP come from. So it will take that long strand of DNA and cut it into sections using restriction enzymes. And this is because, like we said, one strand of DNA has millions of base pairs. That is too many base pairs for scientists to look at. It's too complex. There's too many factors that could be different from one person to another. So if we're looking at the DNA molecule as a whole, that is not going to be considered individual evidence because a lot of our base pairs are going to be the same from one person to another because those are the things that make us human. What we can do is we can look at certain chunks and fragments of that DNA and characterize those pieces. And we look at the likelihood of other individuals having those same pieces. So forensic scientists use DNA fingerprinting to match the unknown samples of DNA found at a crime scene to known samples of DNA in the cells of a suspect. So we look to see if they have the same DNA markers and could this possibly match up with other people that have these same DNA markers. And like I said, these are used in paternity tests to determine who the child's father might be, because half of the child's markers are going to be from the mother, and then the markers that do not match up with the mother have to have come from the father. So we can see if the father has those particular markers. This does tend to be easier because hopefully the field of suspects is a little bit narrower than in a crime. It might be between like 1 to 3 people that you're testing, versus at a crime scene, we might have 20 possible suspects initially. So in order to characterize and compare our DNA, we said it uses those restriction enzymes. It has to be cut into pieces, and we characterize and look at those pieces. So restriction enzymes are used to cut DNA into smaller pieces, and restriction enzymes don't just cut in random places. So restriction enzymes are designed to recognize a specific sequence of bases, and they are meant to cut the DNA at specific points. So anytime it sees a certain sequence, it cuts at that particular point in time. So for example, and this is kind of shown in the picture, the restriction enzyme of ECO-RE will cut DNA whenever it finds the sequence of GAATTC, and it is coded to cut between the G and the A. So if you'll notice in the picture, what is highlighted in orange is the GAATTC, and it cuts between the G and the A, making our 2 fragments. So anywhere on the DNA strand that it has that same sequence, it will cut in that same place each time. And there are different restriction enzymes. There are hundreds of restriction enzymes that all code for a different sequence. You don't have to know like which ones code for which sequence. But I did want to show you that here are 4 other restriction enzymes. Notice like the cutting site, that DNA sequence is different for all of them, and they cut at different places between them. So we said DNA, for it to be characterized, has to be cut into pieces. Once the DNA is cut into different sized fragments, the fragments have to be separated by electrophoresis. And electrophoresis is a lab procedure. We'll do one next week. But this separates DNA fragments according to size. So electrophoresis setup is shown to the right, and this uses a gel. That's what's inside the box, and it uses a voltage source. The voltage source pushes the DNA through the gel, and what happens is longer fragments cannot be pushed through the gel as much, so they will not travel very far, where shorter fragments are going to be able to travel further. So we will end up with these lines or these bars going across the gel as the DNA travels. So the DNA fragments, like we said, we push them through the gel. They are tiny and very close together, so it's very hard to see them. So we have to make them visible. And how we make them visible is we add a probe. The probes are also coded to adhere to certain fragments. So they will essentially dye whatever fragments we are looking for. And it finds a complementary strand on that DNA. And it actually makes it colored and makes it visible. And by using this technique, scientists are able to observe if there's a new pattern of DNA or if it's a matching pattern of DNA. They can analyze an unknown sample from a crime scene and compare the unknown DNA to suspect DNA that has also gone through electrophoresis. And like I said, we're going to do a lab with this next week using dyes, but using it with DNA, we're able to test multiple samples at the same time, and then we can compare them visually side by side. So this is what the electrophoresis gel or DNA fingerprint will look like on the right. And notice we have our crime scene DNA, and then we have DNA from our victim and 3 suspects. We're going to be looking for matching markers, matching bars. So if you're looking at this, crime scene starts off with a lighter gray bar. That matches up with suspect 2, also has that same lighter gray bar. And if we look at crime scene and suspect 2, those are identical. So they would be from the same person. If this was in the case of a paternity case and we had like the mother and then all the possible fathers, we would look at which markers match the mother, and then we would look at the possible fathers to see which other bands match the kids' bands. So with DNA fingerprinting, there are 4 main steps for DNA fingerprinting. And to remember them, I have the anagram, it's ICSI, is how you can remember the 4 steps for DNA fingerprinting. So the first step is to isolate the DNA. The second step is to cut the DNA. Sorry, I put C twice. So cut the DNA using restriction enzymes. Then we have to sort the DNA. And then we have to identify the DNA. And so we said we isolate DNA by getting it out of the cell, removing all of those organelles. Cut the DNA using those restriction enzymes. Sort the DNA using electrophoresis. And then identifies when we look at the electrophoresis print and match up which ones are identical. That's what it means by identify. So we're looking at those specific bands and those specific alleles to actually analyze that DNA fingerprint. All right, that is where we're stopping today. We're going to pick up with this and continue. Whoops. Sorry. We're going to continue with this on Monday and wrap up the rest of the unit.